Hello viewers, today we will discuss about bacteria classification, occurrence of bacteria and structure and composition of bacteria and how bacteria move with the help of. Today in this video we will discuss detail. First of all, here is bacteria. The topic is bacteria. Bacteria are microscopic single-celled organisms. Means bacteria have single cell and they increase in the diverse environment. It means that bacteria are present in the diverse shape and they are microscopic. They have single cell. In this type of organism, only single cell is present. Now we will discuss what is the occurrence of bacteria, means where the bacteria are present. They can live within soil, in the ocean and inside the human gut. Humans' relationship with the bacteria is complex. Sometimes they lend a helping hand by curdling milk into yogurt or helping with our digestion. At other times, they are destructive, causing disease like pneumonia. Pneumonia is a disease and it is very harmful. Now we will discuss about the structure of bacteria. The structure of the bacteria is based on the relative complexity of their cells. All living organisms are broadly classified into two groups. First group is prokaryote and second group is eukaryote. Pro means before and karyote means nucleus and eukaryotes means true nucleus. The organisms having true nucleus is called eukaryotes. And the organisms which do not possess or which do not have a nucleus or the organisms which do not have the organisms are called prokaryotes. Bacteria are prokaryotes means they do not have a cell nucleus. Entire organisms consist of a single cell with a simple internal structure. Means they have simple structure. Unlike eukaryotic DNA, which is neatly packed into a cellular compartment called the nucleus, bacterial DNA floats free in a twisted thread-like mass called nucleoid. Bacterial cells also contain separate circular pieces of DNA called plasmid. In their body, DNA is also circular form present, which is called plasmid, and on the other hand, bacteria lack membrane-bound organelles, specialized cellular structure that are designed to perform a range of cellular functions from energy production to transport of protein. Both bacterial and eukaryotic cell contains ribosomes. It means that bacterial and eukaryotic cells have ribosomes. Both have ribosomes. Bacteria have also ribosome and all the eukaryotic cells have ribosomes. On the outside means on the outside of the bacterial cells. Bacterial cells are generally surrounded by two protective covering, outermost boundary or cell wall and inner cell membrane, and the second is inner cell membrane. However, certain bacteria like the Mycoplasmas do not have a cell wall at all. Some bacteria may even have a third outermost protective layer called capsule. So it means that some one bacteria may have outermost boundary which is called capsule, but the mostly bacteria have surrounded by cell wall and cell membrane. Bacterial surfaces can be covered by whip-like structure or extensions, which is called flagella or pili. Long flagella aid or helps in the motility and movement, while on the other hand, short pili help bacteria to attach on the host surface. Now we will discuss classification of bacteria. A few different criteria are used to classify bacteria. They can be distinguished by nature of their cells wall, by their shape, or by differences in their genetic makeup. It means that we can differentiate them by the nature of their cell wall, by their shape, structure, and differences in their genetic materials or makeup. Gram-Statin is a test. Now we are going to discuss what is gram-positive bacteria and what is gram-negative bacteria. Gram-Statin is a test used to identify bacteria by the composition of their cell walls. It is named for Hans Christian Gram, who developed the technique in 1884. Bacteria are first stained with a purple dye called crystal violet, which specially binds to peptidoglycan, a complex structure of amino acid and sugar found in the cell wall of the bacteria. This is followed by a series of steps that ultimately remove any unbound or loosely bounded crystal violet. Then the cells are stained with a second red colored dye called safranin. What is called safranin? Cells of these bacteria or the bacterial cells are stained with a second red colored dye, which is called safranin. Gram-positive bacteria stain purple because their cell wall are rich in peptidoglycan. On the other hand, gram-negative bacteria, whose cell walls have two layers, take on a red coloring. Outer layer of lipid does not bind strongly to crystal violet, and the dye is easily washed away during the staining process. It means that, for example, we can see that Streptococcus pneumoniae, which cause pneumonia, which is gram-positive bacterium, while on the other hand, E. coli or Escherichia coli and Vibrio cholera, which cause cholera disease, are gram-negative bacteria. Now here we will discuss about the shapes of the bacteria. There are three basic and most popular bacterial shapes are three: round bacteria, cylindrical, and spiral. Cocci can also associate with one another in the different configurations, combinations of two or Diplococcus, a linear chain or Streptococcus, and a cluster or Staphylococcus. The shapes and configuration of the bacteria are reflected in their names. For example, we can say that the milk-curdling Lactobacillus acidophilus are bacilli, and on the other hand, pneumonia-causing Streptococcus pneumoniae are the chain of Cocci. Here you can see in the detail shapes of bacteria. cocci, which are diplococci, and the pneumonia causing it cause pneumonia, and staphylococci it cause other disease. And on the other hand, bacilli bacteria, bacillus anthracis, and vibrus spirilla spirochates. These are the main and most popular shapes of bacteria. Phylogenetic classification. It became possible with the start of nucleotide sequencing technology. Ribosomes are present in all the living organisms. One can look at similarities and differences in the RNA sequences that encode certain ribosomal protein and determine the degree of relatedness of different organisms. So it was today our lecture. Thanks for watching.